Namaskaram everyone. My name is Nikhil Mishra and I am doing my post-graduation in food technology from the Department of Dairy Science and Food Technology in Hindu University. Let's talk about nutrigenomics in detail in this presentation. By the end of this presentation, you will learn how it what is the origin story behind it, its definition, the tenets, links to chronic diseases, human genome health, polymorphism, gene diet interaction, and a case study which I will be dealing at the end. Now let's talk about its timeline. In 1990s, what happened was the Human Genome Project officially started. What was this project about? This project was about DNA mapping and the sequencing of its base pair, how it works, how it replicates, and all. In 2003, however, the Human Genome Project officially ended. See, this project took 13 years complete, which show how intricate and how long your DNA is. Okay, in 2001, Pellegrin coined the term Nutrigenomics, and in 2007, interrelationship between food and genome was established. Now, origin story is pretty interesting. In 1980s, they did not knew the concept of nutrigenomics, but they were treating phenylketonuria, which is a genetic disorder, lactose intolerance, again a genetic disorder, using food as a control method. In phenylketonuria, you are not able to digest protein with efficiency, and same goes with lactose intolerance. You are not able to digest lactose. Now, how are you going to define it? It is a study of how food affects a person's gene and how a person's gene affects the way the body responds to food. Okay, nutrigenomics is used to learn more about how genes and diet together may affect a person's health and the risk of developing diseases such as cancer. It may also help find new ways to prevent and treat diseases. This is very important. Okay, or you can define it as in other way or in other words. New science of nutrigenomics teaches how specific food tells your gene what your food tells your gene. What you eat directly determine the genetic messages. It is in form of amino acid sequences, which later will go into transcription and translation, which will form protein, and protein is a precursor of enzymes. And all your body system, all the biochemical reactions happening in your system is due to enzymes. The message in turn controls the molecules that constitute your metabolism, the molecule that tells your body to burn calorie or store them. You can learn the language of your gene and control the message and instruction they give your body and your metabolism. You can radically alter how food interacts with your body and lose weight and optimize your health, which is highly demandable, and it is very appreciable that you are in healthy state, physically and mentally. What are its tenets? Uh, diet-related diet-related genes related to onset, severity, and progression of diseases, improper diet and risk factors, dietary chemical and alteration of genomic structures, and what is the correlation between genetic factor and diet. Now, in order to understand how diet affect your gene, we need to first understand a concept known as central dogma. What is this concept about? In this, you have a DNA, then it will undergo RNA formation through a process known as transcription. Okay, and this RNA is ultimately going to get translated to protein. This phenomena is called transcription and this is translation, and this will further undergo modification to secondary, tertiary protein and ultimately to enzymes. Okay, so all how this enzyme is going to be made is all its information are in your genes. If the factors from food come to contact with gene, they are going to get it altered and gene expression takes place. Okay, if this factor which is interacting with gene is positive and as it may result in toxin destroying enzymes. It is believed that cauliflower, broccoli, and Brussels sprout can regulate gene expression. Now, nutrition gene interaction. There are three type of interaction. Okay, first one being direct interactions, epigenetic interactions, and genetic variation. Now, in direct interaction, nutrient after interacting with the receptor behaves as transcription factors that can bind to DNA to induce expression. What are these transcription factors which are going to take part in transcription and hence forming the mRNA, which is required for protein synthesis? In epigenetic interaction, nutrition can alter the structure of DNA and ultimately gene structure is altered. Let's suppose you have a sequence A, T, G, and C. Here A stands for adenine, this stands for thymine, this is guanine, and this is cytosine. Okay, we have a base pairing rule in the genetics, which is Chargaff rule. Here the A is going to interact or bind with the T, and G is going to interact with or make hydrogen bond with C. If let's suppose the nutrition here going to get alters this, then your mRNA sequence is changed in transcription and hence the protein sequence is changed. Okay, genetic variation, it is mostly single nucleotide polymorphism that can alter expression or functionality of gene. What is polymorphism? It is the presence of two or more variant form of specific DNA sequence that can occur among any different individual of a population. For example, if this T gets replaced with A, you are going to get a whole new kind of protein. Now, its links to chronic diseases, which is very important. ENO, that is European Nutrigenomics Organization, says the diseases are because of the are inflammatory, metabolic, physiological, or oxidative in nature. Okay, now we know that CVDs, that is cardiovascular diseases, obesity, and diabetes is disease of civilization. Soloway says nutrition availability provide selective pressure for genetic drift, and Lynn Ferguson, food intake influenced by gene variant and encoding taste receptors. Further, diet gene interaction are highly complex and hard to predict, thus demonstrating the need for highly controlled genotype and environmental conditions that allows for identifying different regulatory patterns based on diet and genotypes. Now, let's come to the CVD. It was observed in the study that individual with E4 allele in an apolipoprotein E gene shows higher LDL with increased dietary fat intake compared with E1, E2, and E3 allele. Okay, there is also a single nucleotide polymorphism at this position, that is minus 75 G by A in apoprotein A1 gene in women is associated with increased HDL levels. Variant A variant shows increase in productive HDL level of PUFA. Risk of cancer, most dangerous disease of all. Risk of cancer is increased if there is gene-diet interaction between mutated gene. Carcinogen, what is this statement talking about? Carcinogens. The carcinogens may be of chemical nature, it may be of radiation, it may be in the form of radiations. Okay, carcinogen metabolism affect polymorphism and affect the probability of contact between carcinogen and target cell. If this my carcinogen is going to interact, let's suppose this dot here represent a carcinogen, and this is my target cell, where if this interact with this cell, this is going to get it is going to lose its proto its protoconcogenes are going to lose its ability. Okay, then it is going to divide in various cells, leading to form, which will lead to form tumor, which will ultimately lead to formation of cancer. And there was high risk of colorectal cancer with consumption of red meat. Genome health. Okay. How it is defined? Expression, physical integrity, and stability constitute genome health. Okay. There is increasing evidence that genome's instability in absence of overt exposure to genotoxicant is itself sensitive to nutritional deficiency. Folate is critical for genome stability. It is recommended that if you have this much amount, that is 200 micrograms per day, then your genome is going to be exceptionally stable. Folate, vitamin B12, niacin, vitamin E, retinol, and calcium are associated with reduced DNA damage. Now, polymorphism, as I told you earlier, it is the presence of two or more variant forms of specific DNA sequence that can occur among different individuals or population. How it happens? Most genes have small sequence differences. Polymorphism, this if your A, let's suppose in a sequence your A gets substituted with T, then there is going to be a large repercussions of this small change. Okay, this will change your metabolic response to diet and influence its patterns. SNPs can change basic information. Different quantity or structured protein is transcribed. SNP that alter the function of housekeeping gene, involve basic maintenance of the cell, and are assumed to alter risk of developing disease. Dietary factor may differently alter the effect of one or more SNPs to interact or decrease the risk. Gene diet interaction. 97% of genes are associated with human diseases are monogenic. Okay. Now, if you are going to modify your diet, it can prevent some monogenic disease. In PKU, phenylalanine or high protein food should be avoided. In defective dehydrogenase activity, it is again a genetic disorder. You must avoid alcohol. In galactosemic patient, they must avoid milk. Then I will discuss the case study. In this case study, there was a bacteria known as Perillus biolactus. It was used to find biomarkers, and this research is the preliminary result of microassay and subtractive hybridization. Okay, so I talked about ideal marker. What is the quality or the definition or the attribute of ideal marker? Ideal markers would be highly expressed, manifested in the first generation within one developmental stage, and consistent over many generations. Meaning, if you have some marker in your gene, it is going to easily be manifested or expressed in the other generation, and it is going to be stable to very other for the future, more future generations. Okay, so this was it. These are my references. Thank you very much.